Good afternoon. Welcome to the last session, which is composed of a talk followed by panel. So to chair the first of the last, I would like to invite Dr. Bruno Olshausen, who's an associate professor of vision science, optometry, and neuroscience, and director of the Redwood Center for Theoretical Neuroscience at UC Berkeley. He holds PhD from Caltech, and he has been a postdoc fellow at MIT and Cornell. He serves on the editorial board of Journal of Computational Neuroscience and was chair of Gordon Research Conference on Sensory Coding and the Natural Environment in 2004. He has co-edited a book, Probabilistic Models of Perception and Brain Function, and in last October of 2005, he organized the inaugural Redwood Symposium, which actually really inspired us in many ways, more ways than one. So I'd like to thank him, and I'd like to welcome him to stage.
Well, thanks, Dharmendra, for putting this together. The program which Dharmendra referred to that we held last fall is going to be held on an annual basis, and I'll just put a plug in for that now. That we're going to hold another meeting next fall. We're putting together the program for that now, and that will be on our website at redwood.berkeley.edu in the next month or so. Well, it's an honor to introduce Christof. This is actually the second occasion that I've had to introduce Christof. The first was when I was a faculty member at UC Davis, and Christof came to visit. And I described Christof as a scientist with an incredible childish attitude towards his science. And a lot of people came up to me afterwards and laughed at my, my mistake, saying, "Oh, you meant to say childlike." Of course, maybe yes, that is what I meant to say, but those of you who know Christof will know that there's a fine line between the two. So the talk the stuff that Christof will talk about today is his studies of consciousness, but the research in his lab really covers a very broad range of areas. One is in vision. Folks in his lab have been involved in designing analog VLSI systems for guiding robots, and doing psychophysical studies of visual attention, developing computational models for how attention is deployed in natural scenes. And most recently, together with Gabriel Kreiman and the neurosurgeon Itzhak Fried at UCLA, he's come up with very intriguing evidence for these neurons in the medial temporal lobes, which look everything like grandmother cells, and so very intriguing finding. Another area of research in his lab is in the biophysics of computation. And this is the area where I think Christof really first developed his reputation, long before he became known as that wild and crazy guy with the orange hair who talks like Arnold Schwarzenegger. But he, back in the early 80s, he did some very nice work exploring the biophysical properties of dendrites and showing the kinds of nonlinear computations that can be formed in dendrites, and the kind of information processing capabilities that can occur in single neurons. And this work is really beautifully summarized in a book published by Oxford University Press, Biophysics of Computation, Information Processing in Single Neurons. He's an author of another book, which will cover most of the stuff he's talking about today, The Quest for Consciousness, which recently came out. And I should say, when I was a student at Caltech, Christof really loomed very large in the faculty there. And all the students always wanted to work in Christof's lab. It was always known as the fun lab on campus, and I think after today's talk, you'll see why. So help me welcome Christof.
All right, thank you very much, Bruno. Are we live here? Yeah. All right, so I'll talk to you, as Bruno said, a little bit about the existing work and then about some future work that we would like to do to try to understand consciousness. So first of all, just following what Jerry Edelman mentioned this point briefly, one has to distinguish when studying consciousness two different concepts of consciousness. One is the different states of consciousness, like right now, nobody's asleep, at least not yet, so you're all awake. So that's one state. When you're asleep, you're in a different state. When you're dreaming, you're conscious, but yet again a different state. And the state of consciousness that you have when you dream is quite distinct from the state of consciousness in that when you're awake. So for instance, you don't have you don't have access, you don't transfer things from short into long-term memory. You don't have access to, you don't introspect, etc. And then, of course, the clinical relevant states like MCS, minimal conscious state, and PVS, persistent vegetative state, like Terri Schiavo You remember her case, terrible sad case last year. So those are different states, and this usage of states a little bit like in states of matter, gas states and plasma states and liquid states. And here there are certain neuronal factors that people are interested in, particularly from a clinical point of view. Those involve structures in the midbrain, in the midline thalamic nucleus, for example, the intralaminar nucleus. These are responsible. You have to have certain enabling agents, and if they're not present, you may be in coma or you may be in deep sleep, so you're not conscious at all. Most of the research that's ongoing concerns itself with the content of the current state of consciousness. So right now, your state of consciousness should be filled either by my voice with its teutonic overtones, or it should be filled with by the yellow writing or by the content of what I'm saying. And if suddenly you remember, oh darn it, tomorrow there's a grant deadline, then your content of your current conscious state will shift towards, and remembering what you have to do in order to make that deadline. This is much easier to study than this experimentally. This we can manipulate now with very high precision, both temporal precision, fraction of a second, as well as a fraction of a degree of visual angle. Well, this is much more difficult to modulate precisely. Yes, it's true, each night we go into different states of consciousness as we go normally from awake into sleep in different states of sleep, but all sorts of things happen when you go to sleep. You have atonia, you don't move, right? You don't have access to memory, your blood pressure changes, your memory changes, so all sorts of different things change. Well, the advantage of these things here, I'll show you, I think at least one illusion, everything else change, everything else remains constant except what you're currently conscious of. All right, well, what is it we can say for sure when it comes to consciousness? Well, we can say it seems to be associated with certain types of complex adaptive networks with massive feedback. There's no evidence that in feed-forward networks you will have any conscious sensation. In fact, there's quite a bit of evidence to indicate the contrary, that if you're just in a feed-forward mode of operating, you get what we call a zombie system. You get automatic behaviors without any conscious sensation. Now, it's only true that for some systems that are shaped by natural selection, you get consciousness. For instance, your immune system is very complicated, is highly adaptive, can learn things, can remember them for your entire lifetime, but we don't have any inkling that there is an immune system. It does its work in phenomenological silence, as it were. Down here in our gut, all of us have on the order of 100 million neurons, and you don't want to know what's going on there, right? And in fact, we don't really have access, or we only have exceedingly limited access to what's down there in our gut. So why is it that we have access to certain parts of our brain? Not all. We probably don't have access to the cerebellum. We may not have access to states in the retina. And so the question is, what are the neuronal structures that directly give rise to consciousness? And more specifically, what are the minimal mechanisms in those structures that give rise to consciousness? We believe many species share certain aspects of consciousness with us. That's not to say that the consciousness is the same. But if you look at dogs and monkeys and apes and mice, certainly all mammals, by their behavior, by the similarity of the structure of the nervous system, and by their continuity in evolution, we assume that they're conscious. If I take a little cubic millimeter of a monkey brain, a mouse brain, an elephant brain, and a human brain, nobody except a few neuroanatomists on the planet can really tell the difference. Yes, they're bigger. But of course, our brain isn't the biggest. That's whales and dolphins. But otherwise, if you just look at the hardware itself, it's remarkably similar. And of course, we have the problem that philosophers call the first-person account. I don't even know whether you're conscious, right? You could all be zombies. Now, I assume, I'm not a solipsist, I assume that, A, because it would be exceedingly unlikely on probabilistic ground that I would be the only person who's conscious. But more to the point is, I know because your behavior is so similar to me, because your brain is almost identical to mine, because we're all very closely evolutionarily related, that you have the same internal states that I have. And you can make the same argument with other creatures. Monkeys, they don't quite look like us, they don't talk. But of course, babies don't talk. A fetus doesn't talk. A young baby that's in pain doesn't talk. And a man who's, a patient who's had a stroke doesn't talk. Yet these other systems, organisms, the baby or the fetus or the patient, we still assume still has conscious states. So from a biological point of view, it makes a great deal of sense to assume that at least all mammals, maybe other animals as well, it's very difficult to tell. As you get further and further removed evolutionary from humans, it's more difficult to make these arguments by analogy. So we don't know, we have no idea, I mean, people don't talk about this, but we have no idea what the minimal complexity is of a brain necessary for there to be consciousness. If you look at bees, bees, Srinivasan, for example, in Australia, does this beautiful experiment where you have a Y branch, where at the entry of the Y branch, the bee flies in and sees a red light. And then at the branch point, one of the branches has a red and one of them has a green light. And you can train the bee very quickly, or possibly even quicker than a monkey, in what's called delay match to sample or delay non-match to sample, where the bee will fly in the maze, realize it's red, and then realize, okay, now I have to leave by the red exit. And then you can stagger this. You can stagger these mazes, and so you have 10 of these decision points. At each point, it knows, okay, I saw the red, so I always have to take the red. Or instead of having red, green, you do vertical, horizontal. Or you can generalize. You can go from vision to smell. Or it can do non-match to sample, where it remembers anything that's not green. I always have to pick the branch point that's not green. That's really a very complicated behavior. And so I don't know, since I realized this, I don't kill bugs anymore. Because we just don't know whether it doesn't feel like something to be conscious. For the same reason, I also turned into vegetarian. Anyhow. And of course, what this is a realization that came first to us by Philosopher, Schopenhauer, and Nietzsche, and later on by Sigmund Freud. Most of our behaviors, not just high-level behaviors, most of our behaviors occur in the absence of conscious sensation. This includes things like talking, right? I have no idea. I just have no idea how sentences are formed inside. It's not that I say, okay, this is what I want to say. This is a noun, this is a verb, and then I translate it from German to English, and then I send it to my larynx. I just have a vague idea, and then next thing I hear these words come tumbling out of my mouth. And that's true for almost everything. I open my eyes, and immediately, within a fraction of a second, I see you. I don't have access to the color stages. I don't have access to the edge detection. I don't have access to any of these stages. I have access to this beautiful design interface, much better even than a Macintosh interface, believe it or not. Beautiful interface that seamlessly and that hides all these computational functions that hides from me. So the point is, I probably only have access to a very small fraction of the structures in my brain. And so this is what Francis Crick and I have been advocating since many years, to focus specifically on what we call the neural correlates of consciousness. What are the minimal mechanisms that are jointly necessary for anyone that are necessary for any specific conscious content, like seeing blue or smelling mom's apple pie. So we tend to emphasize local properties of neural circuits without neglecting what Jerry Edelman, Tononi emphasized, the dynamic core, the more global aspects of consciousness. And I'll come back to that in my very last slide. So what's the experimental program? Consciousness is not so much a philosophical problem anymore. It used to be one purely, but it's now, as John Searle has emphasized, it's now becoming more and more a purely empirical problem. So one thing we need, just like a Turing test, we need a Turing test for consciousness. Because, of course, there's no direct relationship between intelligence and consciousness. We need an assay. We need a battery of tests that we can use in patients, in babies, in fetuses, in monkeys, in mice, in bugs, in flies, in C. elegans, in Drosophila. We need behaviors that require consciousness in these various animals. And then lastly, we must move, and neuroscience is now in a position to do so, particularly with the aid of molecular biology. We need to move from correlation, from saying, well, whenever I'm conscious of red, this part of the brain lights up. And whenever the monkey sees this green bar, these neurons fire, that's all observation, that's correlation. We need to move from correlation to causation. So let me give you 3 examples. So this is the oldest, while I talk, you should fixate. Right? Just fixate the cause. Can we dim the room lights? Just fixate the cause. This is the oldest technique in psychology. It's called an after effect. Just remember, is it possible to dim them, to turn them off altogether? Just remember what the colors are. Red is in the upper left and green is in the right. Can we also dim these? Do you see colors? Yes? No? Let's try this again. Okay, so just fixate anyway. It doesn't really matter. The cause. Right? So you should be able to see colors here, right? Now these are called, they're not real. This, by the way, belies the naive notion, what some people call naive realism, that there's a simple one-to-one mapping between the outside world and the inside world in the privacy of our head. Because here, as you see after a while, there is no color really there. It's gray, yet you perceive these colors very vividly, albeit only for a brief time. So now you can, this is another illusion. Can we turn out those spots for this briefly?
No, actually, because we got to see you.
All right, so what you should do is fixate the cause here. Fixate the cause, don't move your eyes, and tell me what you see. All right, so A, as you point out, you should see a big sphere called structure for motion, rotating. But in addition, as at least one person said, you see yellow squares, right? And they should disappear. Otherwise, you should come see me afterwards. All right, one or both of them may disappear. And, you just have to trust me, I'm a scientist. I'm not turning off the monitor here. Plus, of course, you can ask your neighbor and you can check. Oh, thank you very much. Thank you. You can query your neighbor, when do they disappear for her and when do they disappear for you, and they disappear at different times. This is a nice illusion. It's called motion-induced blindness. It's the simplest illusion. It's the one that's easiest to demonstrate in a lecture hall of what we call perceptual suppression, where something is physically present, yet sometimes you see it, sometimes don't. And this is a very simple operational definition of consciousness, right? It's very difficult to define what consciousness is. But here, sometimes you're conscious of the yellow and sometimes you're not. Sometimes you see the yellow, you can talk about them, they remind you of the yellow sun and yellow Van Gogh and all sorts of other yellow things, and sometimes you can't, they're just gone. So where's the difference in the brain? Very simple question. Where's the difference in the brain between this conscious state and the unconscious state? Now remember the after effect, right? So the question is, will you get... just keep on fixing there. Will you get an after effect for the yellow squares? All right? You should all see the squares, right? And they have an interesting dynamic that we still don't understand, right? They wax and wave. Do you see that? Sometimes it's there and sometimes they're not there. And this becomes sort of weaker. Now, one question is that people, until 10 years ago would have just thought stupid, is do you need to see the yellow in order to get an after effect? In other words, for example, when you compare, let's say you have your range circumstance that for 5 seconds you don't see this yellow square, it's just perceptually suppressed. But for 5 seconds you do see this yellow square. Now you can compare the after image. You can compare the strength of the after image and the duration. You can ask, is there a difference? Okay, we did this clear, very simple, unambiguous answer. There's no difference. Whether or not you see the yellow squares makes absolutely no difference to the strength and duration of the after effect. Why is this interesting? Well, it's interesting because it tells you something. It tells you, A, that you can make progress on the question of consciousness. We're not forever condemned just to talk about it. And furthermore, more specifically, it tells you that in the hierarchy in the visual hierarchy going from the retina to primary visual cortex, etc., the stages where the after effect happens, which is primarily the retina, occurs before the stage where the consciousness happens, where the NCC for yellow squares is. So in other words, whether or not the after effect happens, let's say, in the retina primarily, consciousness for this yellow square probably is in a higher cortical area. So the after effect is totally independent of conscious sensation. Yeah. Not totally, please. All right, who do you see here? All right, that's it. George Carey. Okay. No, don't turn him all the way on because I need him down. Who do you see now? This is pure Bush. And who do you see now? All right, so some of you had the effect. So what happens here is 3 pictures. The first picture is a mixture that we've morphed for one individual, where half the time the individual says, I see Kerry, and half the time I see Bush. The second image is 100% Bush. The third image is exactly the same as the first image. But now, since you looked at Bush for a while, you adapt to Bush, and you're less likely to see Bush. It's not true in reality, unfortunately. So this is called a phase-specific after effect. It's very specific to phases. It's specific to the identity. It's specific to ethnicity. So if you adapt to a white face, then you're less likely to see a white face. It's specific to gender, and it's specific to identity, which implies there have to be very specific neuronal mechanisms in the brain that mediate this. Now, again, you can ask the question. I won't tell you how we do it. It's a more fancy technique. Do you need to see the face in order to get a phase-specific after effect? And we have techniques. Just like David Copperfield, right? You go to David Copperfield shows, he has a beautiful bikini-clad assistant, and he'll make things disappear in front of your eyes, right? Happens very reliable. Now, we can do the same thing in a magnet or in a lab. We don't use techniques like that. We use sort of more boring psychophysical techniques, but we can also make things disappear. You look at them, you don't see them. Just like I showed you the simple illusion, motion-induced blindness. So we can make a face disappear for, let's say, 5 seconds, and then ask you to get a face-specific after effect. Here the answer, the clear, unambiguous answer is you do not. So you can bracket it. So, for example, the after images for colors don't depend on consciousness. The after effect for faces do depend on consciousness. And again, tells you something in the hierarchy, where are the neuronal stages that are responsible for generating conscious perception for faces. So we just perfected this technique. It's called continuous flash suppression. I can't demonstrate it because it requires, not here, it requires that I give you input to 2 different eyes. So what we can do, for example, we can put an angry face like that, which is highly salient. It's angry. It's behavioral, of course, very important. We can put an image of an angry face in one eye. And then if we flash different images into your right eye, like Mondrian's, they always look different, you can suppress. You suppress, and you don't do it consciously, and it's not in any Freudian sense or anything like that. It's just you don't see, in this case, you may not see for 3-4 minutes this face at all. Although physically the image is always present on the eye. It doesn't matter on which eye you do it. Because perceptually what you see is always this. So what you consciously see is always this. So again, you can suppress this face very effective from view, and you can now study. These things are interesting because they allow you to study the footprints of consciousness, as it were. Because now you can compare in a magnet or in an animal with electrophysiology, you can compare where's the difference between the seeing it and not seeing it. In both cases, the physical input is present. This is what I meant with my very first slide. You can really titrate. You can really keep everything constant. My arousal is constant. My memory is constant. I'm always awake. The physical input is always present. But sometimes I see it and sometimes I don't. And so I can use that to dissociate, to look at the brain regions. Okay, so I just published this fun essay in Nature, and you'll see how it relates to the previous slides. So I get bombarded, and you all know it also because you're physicists, almost every day with a new proposal about quantum mechanics and consciousness. And so I couldn't resist writing this thing. So let me just make a brief comment. There's no question that quantum mechanics analyze chemistry, which analyze biochemistry, which analyze biophysics, which analyze neuroscience, which analyze cognition behaviors. There's no question about that. But the relevant question is that people are interested in whether the brain has whether the brain or its component display macroscopic behavior, particular quantum entanglement, that are of direct relevance to the brain. Now, of course, to its behavior. Now, of course, a priori, 300 degrees Kelvin, hot environment, closely coupled to the environment, doesn't sound very promising, does it? And of course, we know because there is this large field of quantum computation, which partly, originated at IBM. We know 2 things. We know practically it's immense difficulty. I mean, there have been hundreds of groups over the last 10 years with a lot of funding. Trying to implement quantum systems. And, the record now is, I don't know, doing a factorization of 15, right, 3 times 5, with 4 or 5 quantum states. So we know, due to decoherence, large-scale decoherence, it's a very, very, very difficult thing, even in these physical systems that you can keep very isolated. Furthermore, we know computationally that the algorithms, particular Shor's algorithm, where you can go from a super polynomial to subpolynomial, they're not very useful to the brain. The brain's job, the brain's daily chore, does not involve doing factorization. Okay, that's not what the brain is all about. What the brain is about is doing things like linear products. If you've seen some of the talks today and yesterday, the rules that people use are sort of linear products, inner products, outer products, thresholds, robust analog operations. These are not of the type, at least, that we have any theory for that you can do much faster in quantum computation. Right? The hardware that we have at the brain. So this is a quarter of a micron. Okay, this is individual synapses here. Okay, so this is the stuff with which we compute. This is a spine, asymmetric synapses, makes a spine onto here. And if you go down to a further level, this is a single channel. And these channels, as far as we can tell, they switch. They are stochastic, microscopic, stochastic switching elements. But these are, of course, they're huge, right? They're 20 or 30,000 Daltons or 50,000 Daltons. So that by any standard, they're very, very big objects, right? And we have no evidence. In fact, we have lots of evidence that what happens if there's a technique called patch clamping. So, for example, you can just record from the current, tiny current through one of these channels, or we're talking about picosiemens here, or one of these channels. You can study the superposition. There's no evidence that if you measure two or more of these channels, that they behave anything than what you would expect from conventional linear superposition. There's no evidence whatsoever for any quantum mechanical effects at the level of individual channels. Yeah. Here you have, this is one of the best known neurons there is. It's from Kevin Martin, Rodney Douglas group in Zurich. It's quite well known because it got published in a couple of hundred papers. It's called Johnny 4. It's from a cat. Layer 5 pyramidal cell, beautiful dendrites. These are the local axons. And again, this is the stuff that, as Henry showed yesterday, you have 100,000 of these all bunched up in this 300-degree Kelvin tissue. So, and the two key biophysical operations that a neuron has to perform. At the input end, it's synaptic input, converting the previous input into neurotransmitter release, and that are then converted back into electric potential. This is a dispersive or diffusive process. It involves release of a few thousand, like 2,000 molecules of glutamate that have to diffuse over the synaptic cleft, separating the pre- and the postsynaptic neuron. Okay, it's really a very bad operation if you want to preserve any sort of quantum bits across that process. Yet this is what brains do. Right? There are roughly 800 million synapses per cubic millimeter, and they all have this dispersive property. At the output end of a neuron, or at the cell body, you have hundreds, if not thousands, of ionic channels that are distributed over space that come together to give rise to a microscopic phenomena that we can capture with Hodgkin-Huxley and other equations to give rise to an action potential. Again, this is a large-scale operation that will destroy any coherent underlying quantum states, if there would be quantum states inside the neuron. So there's just no mechanical or no mechanistic, there's no mechanism how you can get quantum states, even if there's somewhere to be, like, for example, Hameroff and Penrose proposed in the microtubuli, how those quantum states would interact with other information outside the neuron, and then let alone influence the neuron that's from, in my MT area, and then I need to influence a neuron that's 10 centimeters away in the frontal lobe. There's just no evidence for that whatsoever. And then you can really do a cute experiment, which we propose. So remember that technique I just showed you, the continuous flash suppression. Well, you can do this in the following way. You can do Schrödinger's cat experiment, right, where you have a cat, sort of an evil experiment today. I mean, involves killing a cat. So here you have a cat, and you have a live cat and a dead cat, and here you have the flask, and somewhere here you have the Geiger counter that counts whether this one ion will decay, and it decays with probability 50% it'll decay. The Geiger counter will trigger, the hammer will come down, smash the flask, the gas will open, the cat will die. And of course, if it's totally isolated, it's in the superposition of the two states. So let's say you do your continuous flash suppression experiment. So on the right side, you flash these images, not Mondrians, but faces. You flash, let's say, faces at 10 hertz, a steady stream of images comes into your right hand, into your right eye. This happens to be actually the image of Erwin Schrödinger himself. So you have these faces that come in at, let's see, a new face every 100 milliseconds. Perceptually, this will profoundly suppress anything that comes in the left eye. And what you will see is that what's present on the right eye. You will not see anything that's on the left eye. And this may go on for many minutes. So if these people who propose that quantum mechanics is relevant, that you need a conscious observer in order to collapse of the quantum system, that it's not due to large-scale decoherence, the idea would be that for the many minutes that this information is present in my eye, and the photons surely are going to interact with my photoreceptor opsins, right? Yet as long as I don't consciously see it, the system is still in a superposition of states, and it would only lose its superposition, and the cat would either be condemned to be dead or alive once I consciously saw it after many minutes. This to me sounds extremely implausible. Anyhow, so that's sort of a nice Gedanken experiment. Well, let's come to something more concrete, back to two other stories. Mouse and human neurons. So most of what we know about neurons is from animals, 99.99%. The only time we have access to neurons in humans is by and large post-mortem, when the brain is dead, or when you take a biopsy. But there are a few occasions, and this is an occasion with Itzhak Friedus, a neurosurgeon and a neuroscientist at UCLA Medical School, and with, as Bruno mentioned, Gabriel Kreiman is now at MIT, Kian Quiroga is now in England, Leila, she's also now at MIT, he's still with me. And the occasion is that there are epileptic patients, and a subset of these epileptic patients are refractory to pharmacological treatment. The drugs don't work anymore, and these patients will have 1 seizure a day or 10 seizures a day. And then a very successful surgical treatment is to take out, to identify what the foci is for certain types of seizures. It only works for the focal type of seizure. To identify where the foci originates, where the epileptic seizure originates, and then take out that part of the brain using a surgeon's knife. Now, in a subset of patients, you can't do that. You can't locate from the outside. You do X-rays, you do MRI, you do EEG, you don't know. Sometimes you don't even know whether the seizure originates in the left or on the right side of the brain. So then what you do invasive brain surgery. So you drill 8, 10, 12 little burr holes, and you insert macroelectrodes. So you insert these macroelectrodes. These are polyurethane, flexible. They have 7 leads, platinum leads. And you insert these into the brain, here into the hippocampus. And then you can essentially do intracranial EEGs, like EEG outside, except now they're inside the brain. And you can do, they're monitored 24/7, and you can do triangulation to pinpoint where the seizure originates. The patient is now in the clinic for 3, 4, 5, 6 days until he or she has a couple of seizures, and the neuroradiologist can pinpoint it, and then the electrodes are taken out, and the foci is taken out, and it's a quite successful operation. Now, what Itzhak Fried did, they hollowed out this inner lumen, and they inserted through this inner lumen, now through this inner space, they insert these 9 electrodes, 9 conventional microelectrodes. We measure everything relative to 1, so we have 8 pieces, 8 microwires in there, and there are 8 to 10 macrowires, and we now have a 128-channel system. So we can now essentially record from a patient for a couple of days from 100 wires roughly inside the patient's head. And the patient can tell us directly what he says. We have to do a lot of fancy signal processing because it's a very noisy environment. It's a clinic. It's not like inside a Faraday cage, like in a lab. It's a very noisy environment. There's EKG machines and all sorts of other stuff. But we do all of that. And then I'll just talk to you about this one study. It came out a year ago. We found these neurons in the medial temporal lobe. So this is a part of the brain, particularly the hippocampus, most of the neurons. This is a part of the brain that, let's see, if you're going to remember anything tomorrow about today, about my talk today, it's because your visual input and your auditory input go together into the hippocampus, are somehow bound together, and are consolidated there, and that information ultimately is then sent out again to be remembered over the long term, if you're going to remember anything about this talk over the long term, in the rest of the cortex. So those are the neurons that we're tapping into here. And the time scale is here. This is 3 seconds. Can you see the 2 vertical bars? That's 1 second. So the image is shown for 1 second between those 2 vertical bars. It's an image on a laptop computer sitting on the lap of the patient. So this is 1 second before the stimulus, 1 second during the stimulus, 1 second after the stimulus. There's 6 trials, all randomly intermixed. So here's a picture of a spider. The neuron doesn't fire 1 spike. Picture of, what's his name, Kobe Bryant. The neuron fires once, not at all. Picture of these actresses. The neuron doesn't really fire. Here you see Jennifer Aniston. 7 different pictures of Jennifer Aniston. The neuron fires a lot. Very selectively, exactly around 290, 300, 310 milliseconds, the neuron starts firing, which is a very long delay by monkey standards. Here she's married, or she used to be married. Turns out she's now divorcing or has divorced. I guess she's divorced, I think. And here you see pictures of her with her then husband, and the neuron didn't fire. So the neuron here did not fire to when Jennifer Aniston was present with her husband. The neuron only fired to her. This is a different neuron that has a higher maintain rate. This is probably multi-unit. The neuron has a highly maintained rate. But then you can still see superimposed, roughly at 300 milliseconds, it fires, in this case to the Sydney Opera House, and here even to the text, Sydney Opera. Now, in terms of visuals, some of you I know are visual scientists or machine vision people, it's quite remarkable how invariant these cells are. And of course, that's how invariant our memory is. Because this cell will respond whether it's yellow on black, blue on white, white on gray, or when it's the text, right? There's almost there's no visual similarity at the bit level between this image and this image. Yet for us, they both represent Sydney Opera House, and the neuron fires to them. This is another actress called... she played in a Catwoman. So she played in this movie Catwoman. Yeah, Halle Berry. So here it's you can see her in her dress. Here's a line drawing of her. Here there's, you see different pictures of her. Here's another woman dressed as a cat, but the patient knew that wasn't Halle Berry, so didn't fire through the text. On the same neuron, so we have multiple, we can listen, because we do this wavelets decomposition, we can, on one neuron, on one electrode, we can listen to multiple neurons. So from the same wire you saw the previous Halle Berry cell, there was also Mother Teresa cell. Don't know why. Pamela Anderson cell, responds to Pamela Anderson. This responds to two objects. This responds either to this or to these. Here's one to Brad Pitt and Jennifer Aniston. Anyhow, so we have roughly 15% of our cells respond to specific images, and 1/3 of those, i.e. 5%, respond this invariant way. Now, it may well be possible that 100% of the images of the neuron will respond to image, but I only have half an hour time, then the patient is bored, or his mother comes, or the doctor comes, and I have to stop the experiment, right? So that's an inherent limitation. But some of these neurons, or 1/3 of these neurons, are very, very invariant. And you can try to do... So a little bit, I guess, what Les Verlin talked about, you can try to use probabilistic reasoning about the sparseness of a neuron if you make a specific model. So we have a very simple binary model of a neuron, because we like to know, given we show, let's say, 100 images, and given we have 80 neurons that we record from on one trial, the next day we have 80 images, and we have 12 neurons we record from, we'd like to put bounds on the sparseness of the neurons, right, to see how many neurons. What's the sparseness of the neuron? In other words, what fraction of the input images will they respond to? And how we try to estimate how many neurons will there be that code for a particular percept, like Jennifer Aniston. Because we do have to explain the seemingly paradox that, out of all the possible percepts, concepts, psychologists estimate like 20 to 30,000. The average person has like 20, 30,000 different concepts. So you have to ask, what's the probability that during my half-hour experiment I happen to hit just the right cell that just happened to respond to Jennifer Aniston? So you can do that, and then I spare you the details. You get numbers like this, that roughly we get 0.25% sparseness. In other words, the typical neuron will respond on average only to 1 out of 400 different images. And if you put in this number supplied by Biedermann of 10 to 30,000 distinct concepts, and we're roughly recording from 1 billion neurons, if you count the left and the right medial temporal lobe, which includes the hippocampus, amygdala, parahippocampal gyrus, entorhinal cortex, roughly 1 billion neurons, 10 to the 9, you get something that each concept, each of these 10 to 30,000 concepts, is stored in an invariant way in roughly 2 million neurons. So this is a lot of neurons, 2 times 10 to the 6. Of course, it's a tiny fraction of all the neurons that are present. And each neuron represents 20 to 70 concepts. So it's a far cry from a grandmother neuron, right? The grandmother neuron would be, each neuron would represent one concept, just grandmother. This is very unlikely to be true. So these are, in fact, this is not conceptually, if you read Barlow's early paper in 1972, it's remarkable. If you look at the last paragraph, I mean, his numbers are quite different. And of course, now we have data, but it's really close to the ideas that Horace Barlow had about cardinal neurons more than 30 years ago. All right, so let me come to the last part. The need for doing mouse experiments, and that's, I think, where a lot of our efforts will go in the next year. So these are experiments done together with David Anderson at Caltech, Michael Fanslow at UCLA, and 2 postdocs in my lab, C.J. Han and Colm O'Toole. So the idea what we want to do, because ultimately we want to move to perturbation techniques. We want to take advantage of molecular biology to perturb the system, to enact, we'd like to understand consciousness at the circuit level. In order to do that, we need to specifically, deliberately, transiently, reversibly inactivate and activate different sets of neuronal population. I like to toggle off all the groups of cells that sit in layer 5A in a monkey, and that just those neurons that project to the claustrum, for instance. I want to inactivate this group of interneurons in layer 3, just see those in order to see what's the effect of that. That really requires a molecular technique. So we have to do what people in medicine do, or lots of other areas in biology. We have to develop a rodent model of consciousness. And once again, we're not saying that mouse are conscious as we are, but we're saying they share some aspects of consciousness. Now, they're not very visual. They have eyes, they have visual cortex. It's been very difficult, as Mike Stryker can tell you, it's been very difficult to get mice to do visual things very well. But there's another conditioning paradigm where it works very well. So this is Pavlovian aversive conditioning, the dog and the saliva. This is negative conditioning that we're doing in mice and we're doing in Caltech undergraduate students. So we give tones and shocks. So there are two forms of conditioning. This is a question that goes back all the way to 1923 or so, where people first ask, do you need to be conscious in order to be conditioned? And the answer is it depends. There are two forms of conditioning. I mean, there are many forms. So when we talk about Pavlovian aversive conditioning, where you have a CS and a US, let's say a tone and let's say a high tone, beep, and boop, low tone, and then you get a shock. Let's say only after the high tone, but not after the low tone. Okay, this is aversive differential conditioning. Now what you do, in delay conditioning, there's essentially the overlap in time. So the tone and the shock occur more or less, they co-occur together. So you get the tone immediately, you get the shock. Then there's trace conditioning, because there's a trace period, that's more complicated. So first you have the tone, beep, so the tone may come 2 seconds or 3, the shock may come sometime later. That's more sophisticated, and there's nice evidence from Larry Squire's lab in humans, and also we've replicated that in a different paradigm, as I said, with Caltech undergraduates, that in order to be conditioned under this trace conditioning, humans need to be aware of the CS-US contingency awareness. What that means is they need to be able to say, yes, there were high tones and there were low tones. The low tones never followed the shock, but the high tones always preceded the shock. And if people know that information, then they get conditioned. If I put them in the same context where they have to do a difficult attention-demanding task, and their tones and their shocks, they still get delay conditioning, even if they don't know, well, yeah, there were tones, there were high tones, low tones, there were shocks, but I don't know what the relationship between was them. They can still be conditioned with delay, but not under trace. In order to be trace conditioned, this more complicated form, where you have to put things in short-term memory, because you've got to remember, oh, there was a tone, and then 10 seconds later there was a shock. That seems to require awareness. So can we do that in mice? So this is what we did after many years. It works now very nicely. So you have a tone. Now this is the mouse protocol. Humans slightly different. Beep, 16 seconds. And then you get the shock. And then you have like 3 minutes, essentially you wait, and then you have the next pairing. So you have 6 pairings. Aversive conditioning works very robustly. I mean, I'll show you a movie. It works very well. In trace conditioning, you have 16 seconds tone, then 18 seconds a gap, and then the shock. And then we do this distracting. So we would like to do what Larry Squire did. Larry Squire had people watch a movie, a silent movie. We had people do a difficult task. Well, here we want to do the Murin equivalent, so we get mice distracted by flashing the room lights. You know, we turn the lights up and down with some random reason. Oh, there's no tone. Okay. So that's a tone. Now 18 seconds blank. And occasionally the lights are flashed. So these are 4 different mice being conditioned in 4 separate cages. We just optically feed the image together so you can see all 4. And there you can see, right, it's pretty clear what happened. Now this shows you also a big difference between mice and human conditioning. Right, in human conditioning, the way that we run the protocol, we ask people to themselves adjust the current strength, so the stimulus is painful. Right, and so people, you know, everybody's of course a little bit nervous, and so people put threshold low, and so the trouble is very often we have to do 24 pairing, people fall asleep during this paradigm. Now, you know, these mice, they don't fall asleep. Partly, of course, they have no idea that's happening to them, right? The humans, we tell them, you know, they're after all in a psych lab. Here they don't. I mean, they're scared, which probably explains a lot. While these sort of behavior is so much more robust in animals than it is in humans. Of course, we can also be. We know this from war experience or other trauma experiences. This can be very, very powerful. Can affect you for the rest of your life. In fact. All right, and now we test them. So we do 6 of these pairings. This was the very first pairing. So here you're totally naive mice. They were put in the cage on day 1, then taken out. Day 2, they had 6 of these pairings. And then in day 3, we put them in a different cage in a different room to get away from context-specific effects. And we test them. And you can see 2 of these mouse are from the group that had the distractor, and 2 of the mouse are from the group that didn't have the distractor. All 4 mice were trace conditioned. And you should be able to see, in a second you'll hear the tone, or in 10 seconds, it's very faint. And you should be able to judge by yourself which 2 mice got trace conditioned with the distractor and which were not trace conditioned, which were trace conditioned without the distractor. All right, so you can see the mice here in the bottom, they display what's called fear response, they freeze. And that's how we measure conditioning in this case. We have we videotape them and we score them every 2 seconds whether they're freezing or not. This one, you know, they're freezing also a little bit, but they're freezing much less than these. This is, in humans, we measure skin conductance, for example, typically, or heartbeat or pupillary response. We measure skin conductance, what you measure in a lie detector test. Here we measure freezing. All right, so you can clearly see this is the behavior now. You have delay conditioning, trace conditioning. Here's shock only, no tones association. And the black one is without distractor, and this is with distractor. So you can see here statistically these are the same. Here you get this big drop, and yeah, there's no conditioning to the light itself, you know, to the distractor light. Of course, you have to test that. There's also no inference with context-dependent conditioning. And then the advantage of the mouse is you can now, for example, we took out a part of we know from human studies, where human done in Ray Dolan's lab in London, done associative trace and delay conditioning. We know for trace conditioning, the more complex form of conditioning, a very high-level part of the brain called the anterior cingulate cortex, which is here, part of the frontal lobe, is activated. We also knew that there was also some molecular C-FOS data to suggest the same in rodents. So we made surgical lesions, permanent lesions in here. And then what you can show, those permanent lesions nicely interfere with trace, but not with delay. And we can do sham lesion or V1 lesion. It's specifically the lesions in the anterior cingulate. So finally, what we could show with this paradigm, this is the ACC, what we could show with this paradigm, that just like humans, this was well known, mice can be delays and trace condition aversive. Just like in humans, when you distract the animals using an intentional distractor, in this case vision, you interfere with trace, but not with delay conditioning, also not context-dependent conditioning. And we show that if you remove the ACC by lesioning permanently, you interfere with this task. So now you can, well, at what position are we? You can say, well, we have a task that has some resemblance to a task that in human requires awareness or consciousness. And we can study the basis of that. Let's skip this. So let me come to my conclusion. So how do you want to proceed? Well, so I'm somewhat frustrated doing this now since many, many years. Most of the studies in consciousness are psychophysical studies, like I showed you early on, or fMRI studies. I didn't show you, but most of the work involving the neural base of consciousness involves human work, in particular brain imaging. And that's cool, and we also do it, and that's cool, and you know, you can take normal humans and you can study the neural bases. But you got to remember, hemodynamic response is very sluggish, has a delay of many seconds. It's like low-pass filtering your data with a time constant of 4, 5, 6 seconds. The smallest volume that you can image in a human corresponds probably to 8 to 10 to 12 million neurons. So it's something that's at a very different scale from the scale that the nervous system is at. And in order to understand consciousness, or for that matter, perception or action, you need to understand at the level of neurons. Just like if you want to understand chemistry, you need to know about covalent bonds and ionic bonds and von Neufeld. You need to know about electron shells and all of that. Same thing, to understand behavior, you have to understand neurons and how they interact. That's extremely limited what you can do in humans for obvious reasons. Even monkeys, it's very difficult. They're difficult to train. They're very expensive. They have very long generational time. Therefore, most people study the mouse. You could, of course, make the argument and say, well, why not study the fly, or why not study C. elegans? And some people have argued at least there's attention present in the fly. But the evolutionary gap between us and also the discontinuity in behavior is pretty large. So I personally, I'd like to stick with mammals. So that's why we want to study the mouse, because here we can perturb systems. We can begin to perturb in a very nuanced manner. So we need several things. We need a confluence of we need to know the detailed distribution, not only at the regional level, but at the cell-specific level of individual genes. There are roughly 21,000 or something like that genes in the mouse genome. We like to know where they are. And so that's happening now. In fact, this job is almost done by people at the Allen Brain Atlas. Michael is here, in fact. By people at the Allen Brain Atlas up in Seattle. So it's all online. You can get, they've cut the brain in 25 or 200 micron sections, and you can go to their atlas, or you can just Google them, and you can look online. They have the expression pattern of all 20,000 plus genes, each gene specifically in the mouse brain. Why is this interesting? Well, because this allows me now, with the molecular techniques that are being developed, to say, well, there's one gene that's let's say, it's expressed strongly in cortex, but it's expressed very strongly in layer 6. And there's a second gene that's all co-expressed with this. And so if I can devise like a binary strategy where I have promoters that go to gene 1 and gene 2, and they only express in cells where gene 1 and gene 2 are simultaneously present, and then I can silence those genes. So for example, there's a strategy that just came out of Caltech where people developed, it's called GluCL ivermectin strategy, where people developed a tool that you can deliver either by injection or by viral strategy that hones in onto genetically targeted subpopulation of neurons. It activates a chloride conductance. So for as long as that channel is expressed in these cells, the neurons will be shunted. There's this large conductance, the neurons can't spike anymore. They're silenced. And this stuff naturally washes out after a day or two. So in other words, you have a tool called a silencing tool where I can say, well, if I can identify that this set of neurons is coded by those 3 genes, I can go to them and I can turn them off for 2 days. I can have a mouse that's raised perfectly normal, right? It's not a transgenic animal. It's a perfectly normal mouse, raised normally, and then for 3 days I can turn off. For example, I can turn off the neurons that I think are responsible for transmitting blue-yellow information. For example, in the condylocellular pathway from the retina to the LGN. And there are now these techniques that are becoming available. They have been widely available in invertebrates, particularly in the flies, but they're also now being developed for mammals, so we can silence. The early hope that individual genes will be very specific probably will not turn out to be true. If you look at the Allen Brain Atlas, by their counting, maybe only less than 1% or closer to 0.1% of neurons have an incredible specific expression where they only express, let's say something like the claustrum. So probably we need techniques that can hone in on a combination of 2 or 3, maybe even 4 genes. Then we also have, for example, Ed Boyden at Stanford came up a couple of months ago with a fantastic technique where he took the channel rhodopsin 2 channels of the rod photoreceptor and incorporated it into a reporter gene. So essentially what he can do, he can include a photoreceptor into a specific neuron population. And now if you bombard that neuron population with light, you can turn on that, you can drive that neuron population. So let's say you have a smooth cortex, right, where you can put light on. Let's say you have a specific population, let's say that the one in layer 3 that project to MT, from V1 to MT, okay? Let's say you know that there's a specific gene that's expressed in them. Now what you do, you incorporate this rod photoreceptor, basically that's what it's related to that, into these neurons. So now they all have little photoreceptors. You put light on, all the other neurons, the other 99.5% of neurons, they don't care about it because they don't have this receptor in their membrane. But the 0.1% of neurons that happen to encode this, that all happen to be of the same genetic type, and they all project to MT, you can now drive them with light. You can essentially generate, it's like injecting current, but in each one of them. Right? It's like you had a microelectrode in each one of those neurons, except you can do it in the entire population. So now you can specifically drive just the population that goes from layer 3 in V1 to MT. So those techniques are incredibly exciting to me. Also what you want to do, you want to search, right now there are probably already a few thousand different strains of mice. And a lot of these mice may have already some very specific disturbances of consciousness. That, for example, that they're like zombie mice, that they can do delay conditioning but not trace conditioning, or they can do all the behaviors that we can do without consciousness. And so you really want to do these random mutations like they do at, for example, Novartis Research Foundation, where they look for mutations that affect the blood pressure. But here I'm interested in looking for mutations that search for interference in consciousness. All right, so last slide, what is it we need? So we need robust animal models of consciousness. So we have them in vision using monkey, because there the similarity with monkeys and humans is always very strong. We need to develop in other animals, including possibly invertebrates like bees. We need to identify specific coalitions of neurons that give rise to specific NCCs. And then as I said, we need to inactivate them using all sorts of techniques. Direct electrical techniques, of course. We've been doing that since Penfield, right? The Penfield map that Ramachandran showed yesterday, well, we know that because you go on a patient, you stimulate with a microelectrode. But of course, that's very crude and that's invasive. There are also magnetic techniques that you can do today called TMS. Where essentially you have a magnetic field and you briefly switch on a magnetic field that induces current in the underlying neurons. You can use it in normal people, but it's very crude. It goes over square centimeters. That's why, again, I think ultimately we need these molecular techniques. And finally, what we need is really a principled everything I've talked about is sort of more an opportunistic approach, the one that Francis Crick and I've been taking, that we try to understand, we try to get hints from the neuroanatomy, we're looking at structures like the claustrum, etc. We try to get hints from experiment, what are the crucial elements, the crucial switching circuits necessary for consciousness. It's somewhat opportunistic. It's based on the available tools and techniques and insight into the brain. Ultimately, you need a principled theory of consciousness. We want to understand under what condition is what system artificial or natural conscious? When is the internet conscious? Is the Macintosh already conscious but not telling me? And so what we really need is a principled theory of consciousness in the fullness of time. And the only candidate I see right now is the one that Jerry Edelman and Giulio Tononi are looking. I'm not sure this is the final thing, but the direction they're going in terms of information, integration, differentiation seems to be, to me at least, a very, very promising one. So with that, I'd like to conclude.
So, Christof, that was like a machine gun all the way through for an hour. But one thing that I didn't get is the mutations in the mice. So if I take this bat and I hit the mouse on the head, it's going to go unconscious. And it goes unconscious for many reasons. Because I disrupt the sensory input, I disrupt thalamic processing, neocortical processing. I bet that you could have about 5 billion, 10 million, whatever, mutations that are going to affect, perhaps affect consciousness. I mean, for many different reasons, because they're affecting the sensory processing, because they're affecting any number of different pathways that may actually be supportive for consciousness or have actually nothing to do with consciousness. So I don't really know, and to take out layer 6, yeah, you're going to screw up a lot of processing. But does that mean that it's got anything to do with consciousness?
The idea is always to be, you're perfectly right, it can't be random, it has to be targeted. We're looking for things, for example, the animal can still do delay conditioning, but where it can't be trace conditioned anymore. So in other words, its auditory processing must still be intact, and its autonomic system must still be intact in order to have arousal, but this specific combination of storing information with the CS and then put it in the working memory, combine it 10 seconds later with the USZ, we're looking for that particular circuit. So it has to be very specific. And again, it's a strategy that's opportunistic. Like almost always in biology, we hope ultimately to interfere as specific as possible just with these 3 functions that require consciousness, not with all the other behaviors that do not require consciousness. I don't see how else we're going to do it in an experimental feasible way.
There's one question we got then, Tony. Okay.
Nobody said it's going to be easy.
So you showed us an experiment of 2 yellow blocks disappearing. Is the experiment valid even with one eye closed?
Yeah. This one I showed you, yeah.
It
so
still
that's why I can
disappears.
show it here under these conditions. You can do it with one eye.
And it will still disappear.
This does not require binocular input.
Okay. The other question is, you're looking at neurons. It seems like if I equate it to a studying a computer and a black box approach, it's like I'm connecting oscilloscopes to different parts. I'm not studying the software aspect of it by just looking at signals. Is that an analogy? Or
No,
what
I think
is
it's
the
a very pernicious one. I mean, that's the one I grew up with. That's the one we all grew up with. But there is no software in the brain, right? That just doesn't exist. There are neurons, there are voltages, there's dendritic potentials, there's synapses, but there's no software. There's no universal programming entity. There's no register of things like that. You can use those terms, but I think they're much more misleading, and they will lead you to construct totally erroneous theories.
Conditioning is one kind of programming, isn't it?
Yeah, you can think of it like that.
Okay.
You can think of it like that, but there's no universal programming. There's no programming language around. Program assumes a Turing machine, all those things, and I think those are all very misleading, and they don't really help you conceptualize the real problem of biology. That's my opinion.
Working? Yeah. So that was great, Christof, and I always find your talks very inspirational, and I agree with almost everything you say, but I always get stuck on certain things. So I don't want you to think I'm always like just trying to attack. But when you say on that slide that you accept the reductionist hierarchy and having the fact that chemistry is based on biochemistry and so on, and then you start talking about computational primitives, it seems to me there's an inconsistency there. because the point of the reductionist hierarchy is that there are not computational primitives. And I think we have to face in biology the very real possibility that it is not like the computer for exactly the reason that nobody's gone and built in some fundamental primitives. So can you either tell me what the computational primitives of the nervous system are, or remake the argument without mentioning the word computational primitive?
Well, you could just think of them as biophysical operations that are key to the behavior of the brain, such as synaptic interaction, nonlinear synaptic interaction, such as generation of action potentials, such as delay operations.
But the generation of action potential involves organization of proteins, and proteins rely on quantum effects.
Yes, except there's no evidence for any macroscopic quantum states that are entangled over the time scales and the spatial scales that are relevant to the brain.
I agree with that, but that does not mean that quantum mechanics is irrelevant to the brain.
I didn't say irrelevant. I said there are no macroscopic quantum entanglement that are relevant, that seem to be relevant to the behavior. Plus, it's a prediction. I mean, I may be wrong. I just don't see anywhere in the brain.
Okay.
Like you, I strongly doubt that there's any quantum information processing happening in the brain. On the other hand, the logic that you're using for dismissing it to me seems completely backwards, because you say that you point out all the things that neurons do in the brain, and you say that that they can't do the kind of operations that quantum things allow you to do. Shor factoring and Grover search and so forth. But isn't that the whole point? That if you did use it, you wouldn't use it to do what the neurons can already do. You would use it to do what the neurons can't do. So the fact that it doesn't do that the neurons can't do that kind of operation seems to me irrelevant.
Okay, but on the other hand, we also have now, we're beginning to have theories, for example, of vision that seem to capture a lot of aspects of vision, where we can now, for example, Tommaso Poggi's work at MIT, who has shown that you can now replicate, or you can begin to replicate behavior in real images of real subjects with simple neural network architecture, without need of any fancy stuff. You just have the linear, the usual linear, nonlinear operation with some learning thrown in there. So I just don't see any need. Never mind the physics, just computationally, I just don't see any need for calling on quantum mechanics to do things that we think the brain can do. Again, we may be wrong.
Hi. With the after-image experiment, you're able to show that the conscious perception happens at a higher level somewhere. But with the consciously perceiving Jennifer Aniston, can we say anything about where that is in relation to the hippocampal cell that recognizes her?
Yeah, it's probably before. So as I said, I don't think these are the neural structures that are responsible, the one we record from, for the conscious perception of Jennifer Aniston. That probably happens in fusiform gyrus, and there are almost no seizures in that part of, essentially in the back part of the brain, there are no seizures, which means you have no business being there with your microelectrode. And so we don't record from that. These are the structures where the patient remembers. So in fact, this patient actually worked with, she worked in Hollywood, so she worked with her, and she'd seen her on a daily basis. So probably what happened, she remembered, oh, that's Jennifer Aniston, and that was what we actually saw. That's probably the correlate of, that's also why the delay is so long. It's 300 milliseconds, which is inordinate long if it were a purely visual thing. So I think what we're seeing here is not, it's more the access into the memory, what we're observing, not the visual percept.
Very exciting talk, thank you. I wanted to ask, in the last slide, you mentioned one theory of consciousness, which is Edelman and Tonane, and if you can elaborate a little bit of what you would like to see in a theory of consciousness, or what kind of theory of consciousness would be helpful for understanding or for experiments?
Well, I mean, ultimately, a reductionist theory that explains you see, people always say, well, there isn't a lot of facts about consciousness, but you can write a textbook of everything we know about consciousness and how it interferes and what goes on in patients and in animals and prosopagnosia and catatonia and all these states. So you want a theory that ultimately explains all of those things, and that fundamentally explains, I mean, the central dilemma. And I think well, I mean, I should really let Jerry talk about it, that explains why is it that certain physical system of a certain type have these very rich states that are both integrated as well as highly differentiated. And I think, for my money, this is the most promising theory we have right now. And it's independent of the hardware. So it says, at some point the internet could also become conscious. So it's not just a theory like a dual aspect theory like David Sharma. So in his appendix of his PhD thesis, he's very sympathetic toward dual aspect theories of consciousness. In other words, where you say, well, there's the objective aspect of reality, of information you can measure in Shannon, but then there's the subjective. But he doesn't specify what sort of architecture. I mean, it has a light switch. He would say this also has consciousness, right? Now, that's not very appealing. I want something that's a little bit more quantitative that tells me, well, why is this remote microphone amplifier, is it conscious or why is it not conscious, and how could I test it? So you want a theory like that. And I think for my money, it's the most promising approach we have.
You mean, if I understand, maybe I should ask that later in the panel, but so they provide some very nice explanation in graph theory terms of the integration about the different. So according to such a theory, every model, every graph that has this structure will be conscious?
What? Yes, I think so. Yes.
You mentioned that you had become a vegetarian, but aren't there some theories that say that even plants might have a consciousness? Consciousness? Might be conscious?
Well, there was actually this interesting debate in the 19th century about, you can read it, about whether spinal cord is conscious but not telling us. For instance, what they did at the time, they took a frog and decapitated it, and the frog can still jump. And you can put a little acid on its left side. This is a decapitated, or you do on the left side, and it'll scratch off. It won't just generalize, it'll do specific. And if you were raised on a farm, you cut off a chicken head, the chicken can run around, like a headless chicken, right? But then people concluded, well, by and large, the behavior is too simple, it's too stereotypical, there doesn't seem to be any consciousness. Now, I can't rule it out, my spinal cord is conscious, but there doesn't seem to be any evidence for it. So rather than endless speculation, there has to be sufficient complex behavior. So, I mean, forget about plants, and even sort of simple, single-cell organisms, if they just do very stereotypical things, right now, I see no reason to ascribe consciousness for them. This may be wrong ultimately, but I think right now that's my index for consciousness, reasonable, complex, non-stereotypical behavior that involves online dynamic storage of information. Right.
Okie doke. Well, I think it's time to move on to the next part. Thanks, Christof. And the floor back. So I guess we're going to just move on to the panel discussion. Okay.